recording. Good morning, everybody. Good morning, everyone. Again, good morning, my grade 9, my advisory. We are still in the continuation of our topic. Okay, we are in the genetic engineering. Okay, the genetically modified organisms. So last meeting, I told you that we'll be focusing on the genetic engineering, the genetically modified organisms. Why? Because you can actually relate to this. Sure, in what sense? You're actually eating GMOs. We are surrounded by GMOs. And what is the point? Okay, what is the point of creating GMOs? What's in a GMO? Let's tackle first genetic engineering. This is the process of manipulating genes to biotech or biotechnology. An organism produced in this process is called a GMO or genetically modified organism. So again, GMO in 1973, the first GMO, a bacterium or bacteria, bacterium is singular, bacteria is plural. That is the reason why there is no such thing as bacterias, because bacteria itself is a plural. Okay, it's a plural term. Was produced. Okay, the first genetically modified food. Okay, a tomato is called flavor saver. Okay, was sold in the market in 1994. Okay, genetic engineering in plants aims to produce plants that manufacture in natural insecticides and nitrogen-based nutrients, eliminating the need for fertilizers. So meaning to say that these organisms, okay, in farming, okay, these organisms help us a lot in producing more, okay, more outputs or more products, and of course, with a lesser cost. Why? Imagine there's no need for fertilizers, okay? It resists pests, so most probably, we can actually create more outputs out of this. Meaning to say that there are more productions in a lesser cost. This is the reason behind. Okay. In animals, genetic engineering has successfully produced a variety of transgenic insects, roundworms, and vertebrates. In if you're going to ask me, in Singapore, okay, they don't have mosquitoes there, okay, that are contagious or carrying malaria or dengue. Why? They created a mosquito. They created a mosquito which can actually kill, okay, if mated with, it can actually kill the dengue carrying mosquito or a malaria carrying mosquito. Okay, so most probably they release, okay, a GMO of mosquito that can actually kill those mosquito that can cause diseases. So they eliminated now the mosquito that carrying what? Dengue and malaria. Okay, so those are just few. Transgenic insects, roundworms, and vertebrates. Researchers transplanted human tissues to a mice to give them a human immune system and used this strain of mice to study AIDS and to develop treatment for disease. So again, the purpose of this is for the quality of life, preservation of humanity, and again, to develop medicines out of this, right? But how about the effects of GMO? Like for example, in your fast foods, okay, they are actually GMO. Now, too much of everything can actually harm you, okay? Talking about this GMO, these GMOs has nothing to do with your health issues. But then again, too much of them can actually cause harm. Like for example, too much of eating French fries, burgers, okay, can actually cause what? Cancer, colon cancer. Why? Because you're eating too much. There is no fiber. You didn't actually eat fibrous fruits and veggies. So tendency is it's stuck. Okay, it can actually stuck in your large intestine, causing a lot of bacteria or a lot of bacterial proliferations, or proliferations mean to say a lot of bacteria colony or colonize. It actually colonizes your intestinal walls or large intestine. So it causes what? Cancer, colorectal cancer, for example. What else? Too much of drinking soft drinks. Okay, soft drinks, we can consider this as a product of technology also, because you're using aspartame as the sugar. Aspartame is a product of this came from a chemical which are from the GMOs. Okay, aspartame is magic sugar. Okay? These are artificial sweeteners. So which causes now what? Cancer, major cause of cancer, even your sodas. So that's the reason behind. So let's move forward. Recombinant DNA technology is a genetic engineering technique, under genetic engineering again, that combines genes from different organisms. Okay? Insulin, the hormone that regulates blood sugar, and the growth in have been synthesized using the recombinant DNA technology. Okay. So again, this is the recombination. Let's say it is called recombinant DNA. Recombination of DNA from other organisms with the same species, of course. Okay. Again, recombination of DNA from the same species. Okay. One good example there is the DNA that creates hormone insulin. Okay. Remember that insulin is really important in our body simply because it converts your glucose, okay, into glucagon, okay, which is really important since we don't need a lot of sugar in our blood. We need sugar in our blood or glucose in our blood in a small amount, but too much of them can cause what? Diabetes mellitus. Diabetes mellitus is characterized by too much, okay, too much glucose in the blood, which can cause what? If you have a lot of sugar in your blood, it can cause a serious damage. It can cause, again, a, 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 a failure in your organs, some of your organs, and can cause death, of course. Okay? Sir, nag-atake. Diabetic man ni siya. Okay. Diabetes, again, can actually kill. It can also have a dominant effect with high blood pressure, high blood HB, okay, or cardiovascular diseases. Sir, why is it always associated with that? They're always associated because once you have over sugar in your blood, okay, your pressure also, your blood pressure rises up. And it can cause, again, what? It can cause stroke. You have it right. You heard it right. It can cause stroke. So that is the reason behind. So again, these diabetic individuals, they need to take a medication. They need to take a what? A medication in order for them to create, in order for that medicine to create a hormone insulin to convert the excess sugar in the blood. That's the reason behind. They have maintenance medicine. It could be an insulin shot, okay, in a pen type. We have a pen type in that, or a medicine, a tablet, okay, for the sugar, okay, the high blood sugar in the blood. Let's move forward to sex determination in humans. Okay, sex chromosomes are classified as X and Y. Okay, when we say X is female. When you say Y, it's male because female don't have Y chromosome, only X. A human is composed of 23 pairs of chromosomes. Okay, 22 pairs of chromosomes are called autosomes, which are a non-sex determining chromosomes and responsible for your physical appearance, number one. Number two, your attributes, okay, your behavior, your attitudes. So those are under autosomes. And a one pair of gonosome, which is a sex determinant chromosome. It could be XX or it could be XY. Okay. Again, autosomes also are responsible for formation. Okay. The form of your body. Okay. The kaliwat, it's in the autosomes, right? Sex chromosomes are distinct chromosomes that are involved in determining sex of an organism. Like for example, females have 2 X chromosomes. If there is an absence of Y chromosome, it is usually female. Okay. Whereas males have X chromosome and a Y chromosome. Okay. During meiosis, the male XY sex chromosome pair to separate gametes or sperms. So like for example, if we have a zygote from an egg, usually it is from an egg and from a sperm. Okay. If the female usually donates X, okay, females don't have Y. Okay. Usually females donates X chromosome. And it's up to the male if it donates X chromosome or the sperm donates X chromosome or a Y chromosome. If the X is paired by another X, you can have a female, okay, offspring. But if the X is paired by a Y chromosome, okay, if the sperm donates a Y chromosome, we can have a male, okay, male offspring, right? Half of the male gametes produced contains X chromosome and the other half contains a Y chromosome. Since females have 2 X chromosome, all female gametes or eggs has only 1 chromosome or X chromosome. It can only share or it can only donate an X chromosome. An egg fertilized by an X-carrying sperm again develops into a XX or a female, whereas an egg fertilized by a Y-carrying sperm develops into a XY or a male organism or a offspring. Okay, hence we have the table. The Punnett Square is there. So again, if you're going to look at this, okay, we have male, female, and male. If we're gonna cross, okay, we're gonna cross this over, we can have the result, what, 50-50, okay? This is on sex determination in humans. Usually this is 50-50, okay? But sir, how can you explain those all male, all female? Again, it depends upon the situation. It depends upon the donated sperm, the donated or the donated chromosome of the sperm, right? It depends upon, and of course there are a lot of factors also. Like for example, the environment of these organisms, it can actually affect. What else? The medication that they are actually taking, it can also affect. Okay. Monohybrid cross. Okay, monohybrid cross is a cross between 2 organisms with different variations at one genetic chromosome of interest. Like for example, we are talking about a pod or peas. Okay, are you familiar with the plant peas? Okay, so we're talking about green and yellow here. Okay, green, if you're going to look at this, if it is capital letter G, this is dominant. And again, if it is 2 capital letter G, it is considered a what? Homozygous. Have we discussed this one already?
Yes.
Yes,
Okay.
sir.
I think in grade 8 also. Did we, Rabi?
I think it was one of the final lessons, sir.
All right. Okay, because this is the continuation. That's the reason behind. So I don't want you to erase this in your memory or forget about this. Okay. This is discussed again in grade 8 in the last quarter. And this is the continuation. Okay. Monohybrid cross, again, if it is 2 capital letter G, it is considered as homozygous. Homo means one. So one character trait or one allele. Okay. Only the capital letter G or the dominant. Okay. How about all small letter G? Okay. This is the recessive allele. Okay. It will actually form a yellow. Okay. How about capital letter G and small letter G? It is the capital letter G and small letter G. 2 character traits or 2 alleles in a species. It is called a heterozygous. Okay. Heterozygous. Now, heterozygous has a phenotype or phenotypic or phenotype which is green, or the appearance or the physical appearance which is green, but carrying an allele or carrying a genes for yellow because there is a small letter G. Okay. Talking about appearance, okay, if it is genotype, genotype are the different genes, okay, the different genes of an organism. This is comprised of all the genetic characteristics. That is the genotype. How about the phenotype? You see, phenotype, these are the one you can actually observe. Like, for example, the color, okay, the color of the skin, those are phenotypes. What else? The color of the pod, the hair, okay? What else? The color of the hair, the eyes, the nose, those are phenotypes. The one that you can actually observe. Okay? Genotypes, these are the genes, all the genes classified in an organism, right? Like for example, in this case, I always discuss this one because this is really easy. This is a cross between a yellow pod, because this is all small letter G, and a homozygous green pod. Okay. The results are, if you're going to look at this is heterozygous green. Sure, why heterozygous green? Remember that the green is dominant over the yellow. So as you can see, all the green pods there are carrying a gene for yellow. So the genotypic ratio, okay, if you're going to look at the genotypic ratio, it is all heterozygous green. Heterozygous meaning it carries a what? A gene for yellow. Heterozygous green. And the phenotype or phenotypic ratio, they are all green. In physical appearance, if you're going to check on its physical appearance, they're all green, of course. Right? Do you have any questions so far?
None.
All right. Let's have dihybrid cross. Okay, dihybrid cross is the cross between two different lines or genes that differ into observed traits. Or two what? Two traits. Or two, they have two different phenotypes or traits that can observe. The traits that you had observed. Okay, like for example, round and yellow. Okay. Capital letter R is round. This is again in peas, in plants. We can usually observe plants faster than animals or other organisms. Round, okay, and wrinkled. Okay, round, capital letter R will give us an impression that this is the dominant allele for round, and the recessive is wrinkled, or not round, of course. There's no wrinkled but a round, right? Yellow and green. So let's have these two different alleles or genes. Okay. How can we cross this over? Okay. In crossing over with two different alleles, okay, we need to first cross it by themselves. Sure, how can we cross this by themselves? Okay. Let's have this one. Please do not forget this one because this is really important. Like for example, round, wrinkled, yellow, and green. Okay. Let's cross this over. Okay, like, hold on. As you can see, okay, you can actually see here, round, wrinkled, which are heterozygous. Okay, heterozygous round with heterozygous yellow, or yeah, heterozygous yellow was crossed between a heterozygous round with heterozygous yellow. So how can we do this? Simple. First is you need to cross again, okay, cross this over, okay, by themselves. Okay, to get now the what? The impression of, or the possibility, or possible possible progenies, or possible possible alleles that may come out. Okay, crossing over, let's have this one. Okay, let me share first. Oops, it's less than a minute. Okay. Since it is less than a minute now, we cannot continue this one. We will have the dihybrid by next meeting. And before anything else, I would like you to go to your LMS. Please go to your LMS and answer activity number 1. We need to hurry up. We need to speed up. Activity number 1, which is concept analysis. Okay. Again, activity number 1 is now open. Please go to your LMS and answer activity number 1, which is concept analysis. This will end this afternoon. That's around 5 p.m. Right? I will not open this again simply because we are running out of time.